Hello, everyone.
Hi, Kai.
Is this started? I
Okay.
think we're on the schedule already.
Yeah, I think we were just taking a 5-minute break.
Okay, I see. Yeah. Okay.
Yeah, so just we've got about 2 more minutes until we start.
I see. So since I'm the first one, do you mind if I starting to share my screen?
Not at all. Go ahead.
Go.
Okay, great. That was about 5 minutes. So in the interest of time, why don't we get started here? So welcome everyone to the orthopedics poster session. My name is David Bertrand. I am a PhD student in Dr. Bettina Willie's lab over at the Shriners Hospital. So I will be moderating today. So first things first, just a couple of housekeeping items. So in the chat, you can see that our helpful VP Solution here has posted the evaluation forms for this room. So we have that link available. So the way that the poster session is working in the context of our online format here is it's just going to be short 3-minute presentations. So presenters, please try to adhere to that in the interest of time. And similarly to the previous sessions that we saw, we're going to do half the presenters. So in this case, 4, followed by our question period. So save your questions for the middle. And then we'll end with the final 4 presentations and further questions for those presenters. So without further ado, let's jump right in here. So the first presentation will be Kai Sheng with TLR2 activation-induced senescence phenotype on facet joint cartilage involved in the disease progression of adolescent idiopathic scoliosis.
Hi, hello, everyone. Everyone can see my screen and hear my voice?
Yes.
Okay. Apparently, I shortened my title. So for today's talk, my title is Cellular Senescence in Facet Joint Cartilage of Patient with Adolescent Idiopathic Scoliosis, AIS. So AIS is a complex 3-dimensional deformity that affects about 6 million Canadian teenagers' quality of life, resulting in high socioeconomic burden from the disease management. The reason why the curve initiates and how it progresses remain largely unknown. Consequently, there's no disease-modifying drug exist. Despite there's a disagreement of the causation of AIS, there's a consensus among surgeons and researchers that the AIS spine has lower stability compared with healthy individuals, which makes the patient's spine prone to initiate an inner rotation. And this will be is considered as a priming step for the curve formation. Facet joints are synovial joints that are located at the back of each vertebra, functioning limiting the axial rotation of the spine and ensure the spinal stability. Our lab was the first to report the presence of premature and advanced osteoarthritis on the facet joints on our patients. And facet joint osteoarthritis is known to have a detrimental effect on the spinal stability. A recent study, including ours, find a link between chondrocyte senescence and osteoarthritis. Therefore, for current study, my hypothesis is there's an elevated senescence chondrocyte in the osteoarthritic facet joint in our patients. And this could be the reason of the deterioration of not only the cartilage, but also underlying bone. So collectively impairs the biomechanical function of facet joint and ultimately affect the spinal stability and facilitate the curve progression. The specific aim is to correlate the level of chondrocyte senescence with the severity of facet joint osteoarthritis and subchondral bone quality. I will be covering the material and methodology in each individual result. In result 1, we're using P16 as a senescence marker and perform the IHC on the cartilage from both AS patient and healthy non-scoliotic control. And we find AS patient has significantly more P16 positive senescent cell on the cartilage, and the ratio of P16 positive senescent cell as strongly correlated with the ORC score, which is the degree of osteoarthritis. The higher ORC score is, the more aggressive, more degenerative osteoarthritis is. And secondly, we performed a micro CT analysis of the surgical sample of the facet joints and correlate it the subchondral bone density with similarly with the osteoarthritis degree. And we find lower bone mass and more severe osteoarthritis are significantly correlated in this facet joint from AS patient throughout the spine. And finally, we analyze a pair of facet joint at the same vertebral level. Firstly, we noticed that asymmetrical bone density has been found in these pairs, right or left. And based on the direction of axial rotation, we find that the contralateral facet joints tend to have more severe osteoarthritis. We can see they have higher OA score. And in line with our second result, the contralateral facet joint has a lower bone density. So just in summary, we established an association between more severe OA, a lower bone density, and a higher number of senescent cells in the AS facet joint that is not present in non-scoliotic controls. We believe lower bone density could be a result of an inflammatory mediator produced by the senescent chondrocyte that are affecting both osteoclast and osteoblast ability to maintain the bone homeostasis. And this is what we currently focus on study, is the crosstalk from chondrocyte to this two type of bone cell. I would like to thank to my supervisor, my lab members, and the orthopedic surgeons. Thank you.
Excellent. Thank you very much, Kai. So moving on, our next presentation is Muskan Alad with IL1 Beta Induced Drg neuronal hypersensitivity is abrogated by link N.
Just a second, I'm just sharing again. Can you hear me? Hello?
Yes, we
Hello?
can hear you.
Okay, I'm starting. Hello, everyone. So osteoarthritis is a painful and disabling chronic condition which causes abnormal sensory input to the dorsal root ganglion. One of the key players in development of osteoarthritic hyperalgesia is the pro-inflammatory cytokine IL1 beta, which has shown to alter the ion channels and activate other cytokines as well as neuropeptides. Currently, there is no cure for osteoarthritis, and analgesics do not provide any pain relief. But LINCN is a 16 residue peptide derived from LINC protein, has shown to modulate pain behavior in osteoarthritic murine model. And it has also shown to suppress expression of inflammatory and pain markers in human chondrocytes, as we can see in the graph. So we hypothesize that LINCN plays a direct role in modulating IL1 beta induced hyperalgesia in osteoarthritis. To see the effect in joint, human osteoarthritic chondrocytes were isolated from donor and cultured for Western blotting of NF-kappa B, the canonical signaling pathway of IL-1 beta. The IL-1 beta signaling was increased the activity of NF-kappa B. However, when co-incubated with LINCN, as shown for 1 hour, the NF-kappa B activity was significantly blunted, and it is also quantified in the graph. In order to determine LINCN has direct effect in modulating pain activity, we isolated murine DRG neurons from mice L1 to 5 and cultured these neurons in regular media or with IL1 beta, a pro-inflammatory cytokine in OA, and also in hyperalgesia for 7 days and measured for calcium imaging. So when DRG neurons were cultured for IL1 beta, it showed increased intracellular calcium level. We can see it here and here. But when they were co-incubated by Lincoln, this sustained phenomena was blunted. We can see it here as well as here. So to induce hyperalgesia now, we treated the DRGs with capsaicin, that is we stimulated with it, followed by KCL for depolarization. So in preconditioned IL1 beta neuron, it shows increased intracellular calcium level, but Lincoln blunted the sustained calcium increase again. So from this, we can conclude that Lincoln inhibits the IL1 beta signaling and regulates DRG neuronal induced hypersensitivity by altering sensory pain and can be used in osteoarthritic pain treatment. Thank you.
Excellent. Thank you very much, Ms. Khan. So moving on, next is Chrisanne D'Souza, who will be presenting age-related changes in membrane injury and calcium signaling in osteoblasts and osteoclasts.
Can you see my screen? All works well?
Yep, everything's good.
Okay. So good afternoon, everyone. Today I'll be talking to you about my project, which is age-related changes in calcium signaling in osteoblasts and osteoclasts. We know that as we get older, our bones become weaker. And one of the reasons is that because the response of bone to exercise decreases with age. Bone remodeling and ultimately the maintenance of skeletal health depends on this balance between bone formation and bone resorption, which is carried out by bone-forming osteoblasts and bone-resorbing osteoclasts, respectively. The objective of this study was to examine the effects of mechanical injury on osteoblasts or osteoclasts from young or old mice. So for this study, we took young mice about 10 to 12 week old and old mice about 69 to 73 week old and generated primary osteoblast and osteoclast cultures. Now, intracellular calcium oscillations are one of the earliest mechanosensitive responses. And therefore, to assess these oscillations, we loaded the cells with Führer2, which is a calcium indicator dye. And on the right is a representative recording of osteoclasts loaded with this dye. We then perform mechanical stimulation with a micropipette on a single cell. So here it's referred to as a primary responder. And you can see the calcium oscillations in the primary responder, as well as the non-poked cell, here referred to as the secondary responder. So moving on to the results. In osteoblasts, on the left, you can see a representative calcium ratio, red being the primary responder and black being the secondary responder. On the right is the assessed calcium parameters. And what we found was that the area under the curve, which signifies total calcium, was much higher in younger osteoblasts as compared to older osteoblasts. The signaling radius, which is the ability of the primary responder or the poked cell to generate a calcium response in the non-poked secondary cells, also tended to decrease with age. So now moving on to osteoclasts. Again, on the left is a representative calcium ratio in the primary and secondary responder. However, when we assessed the calcium responses, the trends were opposite in osteoclasts. The amplitude of the calcium curve in the primary responders tended to be much higher in older osteoclasts. And similarly, in the secondary responders, the area under the curve was significantly higher in older osteoclasts. So to conclude, osteoblasts from older animals respond less and stimulate fewer secondary responders. Osteoclasts from older animals respond more and stimulate more secondary responders. And this combination of reduced osteoblast responsiveness with increased osteoclast responsiveness may contribute to diminished bone adaptation to mechanical forces in older animals. Thank you.
Thank you very much, Chrisanne. So moving on, our next presentation is Elaine Hamburger presenting targeting senescent cancer cells using senolytic drugs to prevent breast to bone metastasis.
Good afternoon. Can everybody see my screen?
Yes.
Okay, great. Sorry, I'm just trying to hide the bubble up there. Okay, So this afternoon, I'll be talking about senescence in cancer and how to prevent metastatic disease. So what is senescence and why target this? So basically, senescent cells are cells that are no longer dividing, and they release pro-inflammatory and pro-tumorigenic factors when they accumulate and adapt the senescence-associated secretory phenotype. Now, what this is its secretion of pro-inflammatory, pro-tumorigenic factors, which leads to inflammation, cellular proliferation, invasion, and further blood supply to the tumor. Now, with 1 in 2 Canadians expected to develop cancer, 50% of US patients dying annually of solid tumor cancers having bone involvement, spine metastasis commonly formed from primary breast or lung cancer, and chemotherapeutic stress causing the accumulation of senescence, leading to further metastatic disease, we hope to target these cells in order to reduce the metastatic disease and burden on patients. So we will be using a common therapeutic, doxorubicin, which works on primary breast cancer now in current research as well as in practice. And we will be adding 2 senolytic drugs that target senescent cells specifically, ovanillin and Drg. We hypothesize that by combining these drugs, we will reduce SASP release, hopefully diminishing the size and the spread of the cancer, as well as increasing therapeutic efficacy of the chemotherapeutic. So we decided to use a tumor 3D model whereby we make tumor spheroids and embed it in collagen with surrounding stromal cells in order to mimic the tumor microenvironment and test the combination treatment. Sorry, that was fast. So first off, we wanted to see that in fact, chemotherapy is inducing senescence. So we tried this in both a cancer cell line and a stromal cell line. And as you can see, with the accumulation of the chemotherapeutic, there is an increase of senescence. We repeated this with patient-derived bone cells, as well as spine metastasis cells secondary to breast and lung cancer. And again, we see that the chemotherapy is inducing senescence. Now, we turn to the 3D model. Is it working the way we expect? When we use chemotherapy, is the area reducing? Is the outgrowth reducing? And that is exactly what we found. So we moved on to adding the senolytics, the big kicker. Does it in fact increase therapeutic efficacy? And we saw that it does reduce the area of the spheroid significantly. It reduces metabolic activity and overall outgrowth of the mimicked tumor. So overall, we hope that this model can help us find the proper target combination therapy and move forward to animal in vivo models and hopefully then help patients who, have breast cancer, lung cancer, etc., and then years later end up with cancer in the bone. So hopefully we can stop that from happening. Thank you very much.
Thank you very much, Elaine. So let's move on to a question period for the first 4 presentations. So does any of our audience have any questions for Kai, Muskan, Krisan, or Elaine?
Yeah, I have a question for Kai. So for your facet joints, what levels are, because depending on the level of the facet joint, they may not all have arthrosis. So your data seems to suggest all the facet joints have arthrosis.
Yeah, 1 second. Let me share my screen again. So basically, the scoliosis has a very different subtype. Depends on the subtype. We find that there's a generally because during the corrective surgery, a multiple level of facet joint will be resected. And we find for patients with a severe curvature throughout lumbar to thoracic, they all have osteoarthritis. And of course, the most severe osteoarthritis happen in the thoracic curve and basically near the apex. But overall, they do have osteoarthritis compared with a healthy, age-matched organ donor.
Okay, thanks.
I have a question for Len. Hi, thank you. Great presentation, very nice data. I wanted to ask you, what would be your expectation? What do you think it would happen to the cells when you remove the senolytics? Would this be a treatment that patients have to kind of keep on forever, or do you think it would at some point being possible to interrupt it?
So hopefully it is actually not going to be an ongoing treatment, that it would be so it would be an adjuvant therapy with the chemo, and it wouldn't continue forever. So you would have the rounds of chemo adding the senolytic, and when those rounds are done, hopefully that would just be the solution. So that's why I think the animal models are going to come into play, because then we can see long-term if the senescence starts to come back and if we need to bring back the drug. But the whole point is sort of to prevent those cancer cells from reemerging in different places in the body. So we're hoping that that's not the case, but yes, we have to study that. So yeah, exactly on the line of what we're looking at now.
Thank you.
Thanks.
Any more questions for it? Yes.
Yeah, sure. Sorry. I have a question for Chrisanne. There's very interesting results with respect to calcium imaging and pressure application to the different cells, osteoclasts and blasts. I was just wondering, you saw changes with aging, which is incredible. Was there also changes with, let's say, degree of pressure applied to the single cells? Did you look at that? Like maybe there's like maybe there is a response if you apply more pressure, without completely damaging the cell versus less pressure?
Yeah, so actually I didn't have time to present these results because it was only 3 minutes. I was supposed to do that. So we make sure that we do apply the same amount of pressure to all cells, but we did investigate the degree of injury that the cells exhibit. So depending, and we compared young and old osteoblasts and osteoclasts. And what we saw was that the older cells have lesser injury, or they have more minor injuries compared to the younger cells. And we'd have other researchers, and also our lab has shown that these injuries are important for the cells to sense mechanical loads and then transmit these signals downstream. So when you age, when you have this less injury, the cells are not able to adapt well to the responses as well as the younger ones.
Nice. Yeah, thank you.
Thank you.
I have one question.
Yes, go ahead.
Can I ask Christiane?
Sure.
Hi.
Hi.
Nice presentation. My question is, did you notice any sex difference, like male to female mice, especially for like old mice, like for human post menopause women has more prone to have osteoporosis, and from your senior mice, did you notice any sex difference? Especially in your senior group, did you notice any sex differences?
So that is another aim that we are going to be doing, because right now I'm still collecting a huge enough sample size. And then once I do have that sample size, I can then divide and I can look into sex-specific differences as well. But that's one of our goals.
Thank you.
Thank you.
Okay, thank you very much, everyone. So I think in the interest of adhering to the schedule, we'll move on to our remaining presentations. So next up, we have Ryan Ben Lataifa presenting mast cell homing for enhanced fracture healing.
Can you see my screen?
Yes, and we can hear you as well.
Awesome. All right, so hello everyone. So the title of my project is mast cell homing for enhanced fracture healing. So I'll just jump right in. So inorganic polyphosphates, or poly-Ps, are linear chains of orthophosphate residues linked by high energy phosphoanhydride bonds. They have a dose and chain length dependent physiological effect and play key roles in blood coagulation, bone repair, and also modulate immune cell activity. So at the fracture site specifically, poly-Ps are released in massive amounts from degranulating platelets, and thereon are thought to recruit immune cells to the fracture site, such as including mast cells, which actually contain the largest range of pro-osteogenic growth factors and cytokines. So overall, for this project, we have a couple of different objectives. So first, for the first phase, we want to develop a thermal responsive hydrogel that's doped with different concentrations and chain lengths of polyphosphates. And then as part of the second phase, the objective will be to assess the effect of poly-P on mast cells, both in vitro and in vivo. And our hypothesis is that sustained release of polyphosphates from our hydrogel will increase the recruitment of mast cells to the fracture site and thus increase the release of pro-osteogenic growth factors and cytokines. So for the first part of my project, here's an overview of my methods. So first, we determine the optimal gel composition for physical gelation. And then second, we'll use a dialysis membrane diffusion test to characterize the release profile of polyphosphates from the hydrogel. And then to characterize the viscoelastic properties of the gel, we'll perform a rheological analysis. And then finally, to validate the gel for in vivo use, we'll do a cytocompatibility and cell adhesion assay. So here's just a few of the gelation trials that we did. So the formulation that gelated in the best time frame and the shortest time frame for that matter was a 20% poloxamer 108 and a 1.5% PNVCl gel weight by volume. So here's a video you can see here prior to incubation at 37 degrees. Our formulation is liquid. And then after incubating it, at 37 degrees for 45 seconds, we get a positive tube inversion test, indicating that our formulation has indeed gelated. And here are the preliminary results for the poly-P diffusion test over a 24-hour period. So in the figure on the left, you can see the concentration of poly-P sustained at about 15 micromolar. And then in the figure on the right, this depicts the release profile of our hydrogel formulation. So over a 24-hour period, approximately 40% of our total amount of poly-P was released. This experiment is currently ongoing, so I'll have more in the upcoming. But in conclusion, we're developing a thermo-responsive physical hydrogel with release of a bioactive compound, so in this case, poly-P. And we're in the process of validating this gel for in vivo testing in a preclinical model developed by our group. And this will serve as a biodegradable scaffold for cell attachment and migration, which promotes tissue growth. And overall, if our hypothesis is correct, this gel will serve as an immunomodulatory strategy for enhanced fracture repair in susceptible patients and hopefully circumvent fracture complications. And thank you.
Thank you very much, Ryan. So moving on, our next presentation is Rachel Monk with osteogenic potential of a hematine reinforced monazite cement for bone regeneration.
So first and foremost, I would just like to thank the committee and organizing members for allowing me to present today. And as mentioned, I will be essentially discussing a new 2D material and its implications as a scaffold for bone regeneration. So autografts do remain the gold standard for the reconstruction of bone defects. However, their disadvantages have encouraged alternate avenues. And essentially 2D materials have been a really big hit over the past few decades, especially with graphene essentially coming to the forefront. And in particular, in our laboratory, we actually work with several different 2D materials. And a novel one, hematine, is essentially a bioresorbable ultra-thin sheet of iron and oxygen, by which we are essentially examining its osteogenic potency for bone regeneration. So our hypothesis is essentially that hematine decorated over a 3D monazite cement, which is calcium phosphate, would enhance osteogenesis. So how we actually synthesized this 2D hematine material was we essentially took iron oxide, we applied ultrasonics to it, we separated the material sheets, and then we went ahead and we centrifugated this material and we manually exfoliated the good hematine from the unexfoliated hematine, essentially ending up with this nice mono and biolayer nanosheet. And then we went onwards to essentially make this 3D scaffold. We used a 3:1 powder to liquid ratio using these powders here, which gave us essentially a precursor brushite And to convert this to monatite, we used hydrothermal treatment. And then to get our final scaffold result here, we essentially centrifugated our nanosheets over the exterior surface of it. So when comparing to our control that is seen on the right-hand side here, we can essentially see that there's a big change in the surface morphology of the scaffolds as expected, and we see a nice distribution of the nanosheets over the exterior surface. We then move forward to assess in vitro biocompatibility. So first and foremost, we looked at the cytotoxicity of our 3D scaffolds over a period of 7 days, and we did do it on a pre-osteoblastic cell line. And we did conclude that there was minimal cytotoxicity with our scaffolds, as we had greater than 70% of cell viability at all time points. And then we proceeded to essentially look at an earlier marker of osteogenic differentiation. We chose to look at alkaline phosphatase activity. And then directing your attention to the graph here, we can essentially see this is a 2D monolayer control, just to monitor the cell morphology. And then we move on to our 3D monatite cement, which is our control. And then we moved on to our 3D monatite cement that essentially had the humitine nanosheets on the exterior. And we can see that we have really good alkaline phosphatase expressions that were enhanced. And we also have a really good statistical expression that is noted here in the down right corner. So in conclusion, we essentially demonstrate the feasibility and the simplicity of obtaining this ultra-thin 2D hematine. And although we are currently in the process of exploring 3 additional other bone biomarkers, our preliminary results do suggest that hematine could be the next big scaffold in bone tissue regeneration. So really quick, I would just like to acknowledge my collaborators on the project, especially my supervisors, Dr. Merle and Dr. Harvey, and thank you so much.
Excellent. Thank you very much, Rachel. So moving on, our next presentation is Rashad Aita, presenting neurogenic heterotopic ossification secondary to spinal cord injury, a novel mouse model.
Good afternoon, everyone. Can everyone see me and see my screen and hear me properly?
Yes, everything's good.
Perfect. So good afternoon, everyone. Today I'll be presenting preliminary results in support of my thesis project entitled Neurogenic Heterotopic Ossification Secondary to Spinal Cord Injury, a Novel Mouse Model. So let's jump right in, give you a bit of broad background. So spinal cord injuries severely impact the lives of afflicted patients. And unfortunately, these patients often experience neurological deficits and an increased risk of complications. One increasingly recognized complication of SCI is heterotopic ossification, which is the abnormal formation of bone where bone does not usually exist, such as in soft tissues like muscle and tendon. And currently, HO is conceptualized as an inflammatory and neuronal response after SCI, which leads to abnormal bone formation. And current animal models attempt to understand this process. However, they are suboptimal in the way they employ not some of the most common predisposing conditions leading to HO. And novelty in the literature nowadays in lineage tracing studies suggest a consistent involvement of the musculotendinous tissue as well as their potential to differentiate down the osteochondral lineage. This pushes us to pose our hypothesis that a spinal cord transection to induce an SCI with a simultaneous musculotendinous injury in the knee of the same wild-type mouse would lead to the formation of HO near the knee joint, as shown here. So how will we do this? Perform surgery at day 0 to induce HO with a spinal cord injury at T10 and T11 vertebrae, as well as a simultaneous musculotendinous injury only in the left knee of that mouse, while the right knee will be our internal procedural control. 21 days later, we euthanize and process the tissues for micro-CT scanning, and then we reconstruct 3D models to visualize and quantify the ectopic bone. Then we process and stain these samples for von Cossa to visualize abnormal ossification, as well as safran O to stain cartilage. Our first figure shows radiological evidence supporting the formation of HO in SCI and MTI mice only, while SCI alone does not show abnormal formation of bone. If we look to quantify the HO volumes in both groups, first we eliminate the femur, tibia, and patella in each leg to only quantify the region that has abnormal bone. And we find SCI and MTI shows consistently higher HO volumes compared to SCI alone. Our staining, just to remind you, von Cossa stains bone black and safran O stains cartilage red. We find this a relationship between bone and new cartilage forming outside of the normal skeleton, which highlights a zonal alignment to suggest process of bone formation. So in conclusion, our preliminary findings suggest that we were on the verge of producing a novel and clinically relevant and reproducible mouse model for NHO, and that this mouse model requires the combination of SCI and MTI in mice to induce HO, and that our combination of stains shows this zonal alignment between cartilage and novel bone, which is a key characteristic of endochondral ossification. Finally, we'll be using this model hopefully to recapitulate the abnormal bone being formed as seen in clinic in patients suffering from spinal cord injury. Thank you for your attention. Here are my acknowledgements.
Excellent, thank you very much. So moving on to our final presentation of the session, we have Garji Goel presenting evaluating adipose stromal cell-seeded polyurethane polymers as tissue-engineered implantable ligament substitutes.
Hello, everyone. Sorry. Yeah, so you can see my screen and you can hear me?
Yes, we can.
Oh, okay, great. Hello, everyone. I'll jump in. Anterior cruciate ligament, ACL, is the key ligament for knee joint stability, but it is frequently injured in high impact activities like sports. Due to poor healing capacity of ACL, we frequently require surgical reconstruction. So the most commonly used graft for surgical reconstruction of ACL tear is patellar tendon and hamstring tendon autograft. But these autografts have inferior biomechanical properties and frequently result in variable outcomes like post-traumatic osteoarthritis, stiffness, and meniscal tear. So here we aim to develop a tissue-engineered ACL graft substitute. We propose LAFERM 60 or LAFELT as a scaffold material to be seeded with adipose stromal cells, ASCs. LAFERM 60 and LAFELT are flexible, nanoporous polyurethane polymers whose mechanical properties are being studied here. These material will be seeded with ASCs to study material-driven tenogenic differentiation. In this study, we 3D printed LAFERM 60 and LAFELT scaffold and did tensile testing on them to determine its mechanical properties. We then seeded these scaffolds with ASCs isolated from human adipose tissue and cultured them for 7 days. On tensile testing, we found that ultimate tensile strength and Young's modulus for LAFERM 60 and LAFELT was lower than that of ACL, but percentage strain at failure for LAFERM 60 was considerably higher than that of ACL. We then seeded ASCs on LAFERM 60, LAFELT, and PLA scaffolds. So PLA is a rigid material whose biomechanical properties have been extensively studied by our group. The cell adhesion rate of ASCs on LAFERM 60 was found to be approximately 33%, 55% for the LAFELT, and approximately 60% for PLA. ASCs were found to be metabolically active and viable on all 3 scaffolds after 7 days of seeding. After 7 days, we did gene expression of ASCs on all these 3 scaffolds for tenogenic gene markers, that is collagen type 1, collagen type 2, and collagen type 3, and sclerexis. So sclerexis is an early tenogenic gene marker. It was found to be increased 7-folds on LAFERM 60 and 1.5-folds on LAFELT. As the conclusion, we can say LAFERM 60 and LAFELT has lower mechanical properties as compared to ACL, but both of them showed good cell viability, metabolic activity, and upregulation of tenogenic gene markers. As a future direction of this study, we can co-print LAFERM 60 and LAFELT with rigid materials like PLA to get better mechanical properties, and we can seed them with ASCs for longer time points like 21 days to better understand its tenogenic differentiation potential. Lastly, I would like to thank my supervisors, Dr. Derek Rosenzweig and Dr. Rahul Gauri, and I'd like to thank my audience for paying attention to me. Thank you.
Excellent work, thank you very much. So before moving on to the final Q&A period here, I'd just like to remind everyone once again, the evaluation forms for this session are in the chat. So members of the audience, if you wouldn't mind, please clicking the link and providing your feedback. So without further ado, I suppose let's move on to the last set of questions for our for our final 4 presentations. So does anyone have any questions for any of our presenters?
Yeah, so Ryan, what would degrade the gel in vivo?
Hey, so actually, so the gel can be like naturally eroded because of movement and such, and just physical strain in general. And also the cells, well, we hope, it's our theory that they will chew through this and sort of make their way inside in order to build that osseous scaffold in order to make that callus, like in the bone regeneration phase of healing.
Okay, thank you. I have also a question for Rashad. So Rashad, the model developed is very interesting, and thank you very much. So is that how HO forms in humans?
Well, comprehensive reviews show that HO has a variety of forms, and that, for example, endochondral ossification is just one way it can form. It's most likely a combination of intramembranous and endochondral at the same time, and at various spots where the injury is afflicted. But in the context of tendon injury and muscle injury, it is most likely endochondral predominant.
So, but the way you are developing your model, so which does it reflect in terms of clinical relevance? The way you have explained, which one are you targeting?
So in terms of the ossification process?
Yes, because you have a model, and one of the things that clinicians want is a model to relate to the human condition. And so you explained that there are different ways in the human. So your model, which one is it similar to when related to the human?
So for patients that experience tendon injuries, for example, they endure a process of abnormal bone formation, predominantly that's endochondral ossification. And in our case, we find that endochondral ossification was occurring since we saw this zonal alignment of chondrocytes, and which gradually turns into bone.
Okay, thanks.
Thank you, Dr. Mwale. Are there any other questions?
Maybe I'll ask one question. The question is to Rachel. My question is, I think you mentioned you measured the cell viability for the MT3T3 cell. What's the method you measured the viability?
Pardon, say that again. What was the measure, or what did we use?
What's the method? What's the method you measured the cell viability?
We measured it using Almar blue, and we did fluorescence, bottom read.
So that also measured the proliferation. Did you notice any change of the proliferation, like the cell have grow more cell than the viability itself? Because that's also represent, yeah.
Yeah, we did see that. Yeah, I have those results. I just haven't showed them because of the lack of time. And also we did see an increase in mechanical strength of the scaffold as well, but obviously traits.
But they do have higher viability as well as more the ability for more proliferative.
Yeah.
Is that okay?
Yes. Yeah.
Perfect. Any final questions?
I have a question for Ryan.
Yes.
Ryan, this gel is to like reduce the immobilization time for after fracture, or to like speed up the fracture healing.
Yeah, exactly. So basically, it's taking what an innate like healing mechanism, and we're just trying to sort of boost this innate mechanism by enhancing the migration of mast cells to the fracture site, and hopefully helping with increasing the release of pro-osteogenic growth factors and cytokines in patients susceptible.
Yeah. So what do you think, like how, like from how many weeks it will decrease the immobilization time period, like by using these polyphosphate gel?
That I'm not sure. I'm not sure how we don't know that it does help yet. So we do have to, like, this is what we're trying to see, if by administering those polyphosphates, will it actually increase the rate of healing or speed up that healing process or not. So I'm not sure I can answer that question.
Thanks.
So Gagi, short question. So wonderful talk. So in the literature, people use mesenchymal stem cells with some sort of scaffold, and you are using a different variety of stem cell, and then you are using also a scaffold. So what is different? What's unique about your study?
So what's unique about our study is two things. First is the stem cells. So the most commonly used stem cells for this tendon tissue engineering is bone marrow stem cells, and I'm using adipose stromal cells, which are easier to isolate, and they are more abundantly present and has less trauma. The second thing I'm doing is I'm not using growth factors to drive tenogenic differentiation. So the future aspect of this project will be to apply mechanical stimulation. And so the mechanical stimulation will drive the differentiation process rather than involving BMPs or other growth factors. So that's the special thing about our project.
Okay, thanks.
A quick question, actually, and sorry if you're getting bombarded here, Ryan. It was actually on the polyphosphates. So I know, correct me if I'm wrong, but I believe some of the mechanism in, I think with bone or cartilage was that it was preventing ossification to some extent. I was just wondering if you've speculated on a mechanism of how the polyphosphate could be working in recruiting mast cells.
So that I'm not sure, but I do know that in some, I've seen a couple of papers where they suggest the mechanism for attracting those mast cells is by first making those like proteins that are attaching endothelial cells a little more stretchy, like they're stretching out those barriers. And so there's more vascular leakage. But the other thing is that mast cells, when they circulate in blood, are not mature, but they mature specifically at the fracture site. And so what has been seen with polyphosphates is that they can help with differentiation of cells, and also help with the maturing process in different cell lineages and gene expression, modulating gene expression. So that may be a mechanism, but again, it's a relatively new, newly studied molecule for bone healing applications. So we're not, not sure I can. I don't know if that answers your question or not, but
no, it's fine. Yeah, it's just because it is new. So yeah, of course you're going to get a lot of questions related to it. But thank you. Yes.
Thank you.
Okay, great. Thank you very much, everyone. So some very interesting discussion there. I think if there aren't any further questions at this point, we will wrap it up and break for lunch. So the lunch break lasts until 1:15, and the conference will resume at 1:15 with the remainder of our presentations. So thank you very much, everyone, both presenters and audience members. And we will see you back at 1:15.
Thank you, David.